But yeah, this is the intermediate Fiji workshop. So it assumes a little bit of background that you know how to open files and do things. I'm going to go through processing and thresholding and some of the more advanced topics. So when we do image analysis, one of the overarching things is the images that you take can be quite pretty pictures, but the ones that you do the analysis on are usually, they undergo a fair bit of massaging. So you undergo pre-processing in this pipeline, and then you want to segment and you want to analyze on these segmented ones. So what do I mean by that is the pre-processing is filtering, background subtraction, you know, the gentle steps so that you, when you go to segment the image, it's a lot easier. So segmentation is pulling out the objects that you want to then measure. So you can use thresholding, color thresholding, and things like Analyze Particles to end up with all of these regions. Now, you can manually draw regions, but if you have lots of images, it can be a bit tedious. It's also a way to eliminate any bias by having the software develop and, you know, and capture these objects rather than you going, oh, I think that's one I want to pick. Then once you've got your objects, you want to analyze them. And so that's where you're going to perform measurements on things like the intensity, the area, the shape of the object, how many objects there are, things like that. And so that's the workflow that pretty much all image analysis follows. So for the analysis section, I thought I'd start off with what we want to measure and what you can measure in Fiji, because this is a little bit of a refresher from the first workshop. So yeah, I'm going to start with what you can measure, and then we'll go back to, well, how do you then get stuff to feed into that measurement? So in Fiji, you can measure lots of things. So the microscope images can be qualitative, but they can also contain lots of quantitative data. We can measure things in different ways in Fiji. So some of the common ones include just simply measuring fluorescence as a descriptor-based way of saying how bright our pixels are. So our object that we found has a maximum intensity of, you know, a number. We can count things and describe them. So we can say how many nuclei there are, or how many endosomes or cells or bacteria or crystals were in the image. We can also describe the object by saying, how long is it? Or what is the area of that object? We can also describe how round it is or elliptical through some of the description tools in Fiji. We can also take it to live imaging and dynamic imaging events where we look for changes in fluorescence intensity. So that might be with your FRAP experiments, or if you're performing co-localization studies where you're having local enrichment of something or the uptake of something and where it is, you can measure that through multiple frames over time. And you can perform ratiometric imaging, which is methods like FRET or co-localization, where you want to see relative to one another what they are. So you can perform calculations and get the numbers out and measurements that way. All the different types of quantification are going to require very specific controls and equal imaging conditions for all acquisitions. So you have to treat all the images the same if you're going to measure and compare them. So some of the things that you can measure in Fiji are under the option Analyze and then Set Measurements. So if you click that button at the top, it says Analyze, one of the submenus is Set Measurements. And you're going to get a pop-up like this, and it can do quite a few different parameters that you can measure. Generally, these are all going to require some kind of selection, whether you've drawn a line, a square, a circle, or you've had the program draw around nuclei or identify objects automatically. You generally have to tell it something what you want to measure. Most of the tick boxes are relatively self-explanatory. So if you want it to record out what the minimum and maximum gray value from the pixels within the area, it'll do it. Median is a useful one to give you the median instead of the mean. Shape descriptors we'll go over as well. It lets you describe how round an object is, which is great for filtering the data. And the display label is another useful one, which will then tell you what the image name is in the results window. So, or if you've got a complex data set, it tells you what channel you're on and what slice you're on so that you know where where you are or where you've taken that measurement. So if you're doing lots of images and you just want to process them all, export out that results window, that's a really useful one. Now, there are other ones. It is all documented on the Fiji website. You can come and ask questions, of course, and I can help explain them, but they're the main ones that most people use. Importantly, all of the size measurements will use scale in your images. So if you've taken it on something like the confocals, where you've got the proprietary format, so a CZI or a LIF format, it's going to have the scale embedded. But if it doesn't, and you've taken it on one of the basic fluoroscopes, you may have to put in the scale before you get meaningful results. It'll still tell you an area, but it's going to be an area in pixels. So just keep that in mind. Okay, so I described before, one of the measurements that you can do is the shape descriptors option. And so once you've turned that on and you've got a selection, you can run Analyze Measure, and it'll give you in the results window, things that it's measured with these 4 options here: circularity, aspect ratio, roundness, and solidity. So in this example of the example blob image from Fiji, we can threshold it, identify regions, and then measure them. And we'll go through how to do that in the next few slides. But just importantly, to kind of show you where it's useful, if we look at number 9 and number 10, and then come to the results window, we can see that number 9 is quite high in its circularity. And so area or a circularity score of 1 is a perfect circle, and 0 is not a perfect circle. So it's more square. So then object number 10, which is this elongated one, has a score of 0.666. So you could imagine if you were analyzing bacteria and they were rod or cockeye shaped, you could easily segment them based on that. Or if you've got nuclei in the tissue and you're getting some autofluorescent blobs that aren't round, you could filter based on the shape descriptor output. And so I've put in here what the calculations are if you are interested, but most people just use the circularity measurement. You can also measure lines in Fiji. And so you can do the line scan or plot profile method. So if you draw a line across the image, you can run plot profile, and it will present things like the intensity values along that line. And you can use that for co-localization. What's really nice is that on graphs in Fiji, they have scale as well. So if you want to measure the width of the nucleus, you could draw across here, and if it had scale, it would tell you. Now this example doesn't because it's got pixels, but that's just one of the ways that you can measure things. And that's mostly the same. So that's kind of what you could measure. So you could imagine that you can do these area, shape descriptors, intensity measurements on regions. But how do you generate the regions automatically? Well, that's where we do this pre-processing step. Generally speaking, a normal image is going to have some degree of noise. So if you might be able to see that this image, it's got some backgroundy noise. And when you zoom in, it's not perfect, but we want to smooth that out before we start making our selections. So the easiest way to do that is to perform a filter. And it's a mathematical equation that's formed on all of the images, all the pixels of the image. And it's useful for smoothing and getting edges and background corrections in the image. So when you run process filter, there's a whole heap of options: mean, median, Gaussian filters. You could spend an hour talking about all the different ones. The most common ones that people will use is mean and median. But it'll ask you for a radius. And if you're performing a radius of 1, it will find whatever calculation you've said, whether to find the mean or the median, from this 1 pixel, it'll go out by a radius of 1. If it was 2, then there would be a box here of 25 squares, and it would find, diagonally 2 and average them across, right? Then it updates the intensity values of that individual pixel, and it goes across for every single one. There is a reason why you might want to use a different method. So instead of using mean, you can find the median. The mean is really good at smoothing and blurring your image. So you can see here that this edge of the nucleus is quite blurry now. But if you do a median filter, it's better at preserving a kind of more defined edge, which is good for making sure when you go to do your threshold and then your measurements that you're accurately getting the edge and you're not kind of getting this hazy border. And then the next kind of step that you want to be able to do is you want to make sure you've got a nice even background. So you want to do background subtraction. I would say every analysis pipeline that I write will always have background subtraction, either because the sample isn't mounted perfectly flat, issues with the microscope, it could be tissue thickness, things like that. So always perform a background subtraction. It'll save a lot of time. It can remove the uneven illumination, and that's really good for when you make selections, because the thresholds will, be selected a lot nicer and cleaner if you've got an even illumination or even background. So this is the classic way of, if I can figure out how to, oh, there we go. So how we perform background subtraction in Fiji is using a rolling ball. And what we mean by that is if we take the example background image, uneven background image, you have all these points of light, but you can see that it's darker over here and lighter over here. And so when we draw that line scan, you can clearly see the objects in it, or those disks, but the background changes. And so you could do like a median filter on that image, but what will happen then is you'll end up with a smooth background, but you've modified the values on these disks or objects of interest too much, and you won't have the right kind of analysis results. So what you can do is, instead of just doing a single line scan, if you imagine that you did a line scan across every pixel, you could have this surface plot. And if you invert the image, you end up with something like this, where you have these objects dip down and our background is all in the top here. And if we roll a ball across the surface, we can measure the center of mass of that ball as it changes from one side to the other, but you don't want to have it so that the ball is so small that it falls in the holes. And so what do I mean by that? Well, if we imagine just one little part or one line of pixels, and we've got this ball of a defined radius, it will roll across the surface and we get what this change is. And then we can correct against that change and normalize the curve. So the important thing is that the radius of the ball is larger than the holes. And by holes, I mean the objects that you're interested in. And so we're going to do a worked example, and that's going to be on the first two files. And what we want to be able to do is correct this uneven background. So everyone should have downloaded the files, and we can either, you can have a go at working through it, or I can slowly work through it on my screen, because, as I said, it's a bit weird not seeing everyone in the audience knowing that they've all followed along. But basically, if you open the example crystals image, you want to be able to perform a background subtraction. First, I would say draw a line profile to see it. And then we want to measure the object to see how small it is, and then do that background subtraction. So if I can do that. Okay. So if everyone can see the screen, you want to be able to do like a line across the image. And we can see that there's uneven illumination. If we take the short boundary, we can measure it. And I can see on my results window that the length is about 9. Actually, what I'm going to do is I'm going to do that line scan again. But I'm also going to add that line to my ROI manager so that we can come back and measure the same thing once we've corrected it. So then if we go process, subtract background, we can tell it in here how wide we should go. So it was about 9. If we said 15, we know it definitely won't fall in any of the holes because there might be some variation. If I click preview, I can see it looks like it's doing a pretty good job. Now you want to make sure that you're not hitting the light background by mistake, unless you obviously have a wide field image, bright field image, sorry. You can do, if it's an RGB image like this example one is, you can separate the colors. It's not necessary for this. We don't need to create background. Sliding parabolic just uses a different algorithm, and smoothing's okay for this. So if we click OK, and then I bring up my line again and plot that profile, we can see that the background is now smoothed down. And so, does anyone have any questions with that? There is also the
This is a Brisbane City and Ferny Grove train, stopping all stations. The next station is Fair.
Where's Victoria? So in this real world example, we've got some hyphae. We can do the same thing. We can measure across them and find out, what kind of thickness. About 3. Process, subtract background. Let's do 5. And this one has a, no, so that's the example. Maybe if we be a bit more cautious, instead of measuring, you could measure multiple examples and you'd get something like that where it's now smoother. Okay, does anyone have any questions with that? Nope. Okay. There is the answer slide, which shows the steps that I did. We open the images, we perform a plot profile, and we also did the additional measurement to find out how big, measured the size of the foreground objects, and then did that background subtraction, did it again, and saw that it's been flattened. Okay, so another step that you can do in your analysis pipeline is to perform math on the entire image. And so that's just under process math. And because these images, they're just arrays of numbers, essentially, we can perform mathematical operations on them really quickly. You know, you can add the same amount to the whole image or subtract the whole amount. The subtraction is useful because if you've made a thresholded mask, you can then subtract 254. And so instead of it being 0 and 255, it'd be 0 and 1. And then you can do that kind of multiply that by something else to then just keep the areas that you're interested in. And we'll go through that in a little bit later today. The other thing is when you're doing some fret imaging, you might need to do additional flat fielding and normalization where you divide by the mean of some control image. You can do that here under the divide. So this works on the whole image or just your selection that you've done. And so there's some of the kind of pre-filtering steps that you can do. There are more, but they're the common ones. The next thing you're going to do is try to automatically segment out the parts that you want to measure of your image. And so we'll go through the few different ways that you can do that in Fiji. Thresholding is the main one. It's under image adjust threshold. And so in previous workshops, and as I mentioned briefly at the beginning, you can manually draw areas of interest. If you've got cells and you want to manually trace around them, you can add them. But if you had 50 images with 10 things in each image, it's going to take a long time. And so you can automate it by creating ROIs from a segmentation. And that's done by thresholding. The threshold is, it's just a selection on your image based on the pixel intensities. So all you're doing is you're saying, there's two things in my image, background and foreground. And this is where I've drawn the line to separate what they are. So background values are usually quite low and foreground objects are usually quite high. Now that's in the case of fluorescence, but if you're dealing with bright field, then it's the opposite. Thresholding is usually done on just a single color image or a single channel. There is a very specific way for doing color thresholding, and we'll go through that in a few slides. But a normal threshold is just on a single grayscale image. And the output image is a binary image. It's got two things, background and foreground. Now in Fiji, it gives you the object of the foreground with a value of 255 and the object of the background is 0. And so that's where I was saying before, you might want to subtract 254, and then your object is 1 and your background is 0. And you can do nice multiplications on other images from that. You can change it in the settings, but I would advise against it because down the track you might forget, or you might be dealing with other people's code and it won't expect that. So be careful with changing some of the hard-coded underlying settings in Fiji. Okay, so in practice, all it's doing is it's selecting pixels of a certain value. And so here I've made a gradient image where it goes from 0 to 255. And I've done image adjust threshold and got this little pop-up. And I can move these two sliders to pick things within the range. So if I want to take things from 126 to 255, that's what it would be selecting. If I go and manually move it the other way, I can select just the background if that's what I'm more interested in. If I'm interested in where things aren't and not where they are. Or if you've got a bright field image, you can do that. Now these are text boxes, so you can type it in manually. But in practice, you probably want to do automatically because when you write your methods paper, the method section of your paper, you can't really write, oh, we picked this because that's what we thought picked it, or made that selection. And so you want to use one of the algorithms. So what does it look like in practice and not just on a gradient? So we go back to that nuclei image that we were looking at before with the mean filtering and median filtering. We can take a histogram And we can see that there's a lot of black pixels, and there's some of the lighter gray pixels that make up the nucleus. So all of this is our background, and all of that is the foreground object. What we want to do is tell the software where to make that cutoff point so that we can threshold out the objects. It helps if we do that filtering step, and so we can get rid of some of the noise, and we end up kind of widening or narrowing the background section and putting a wider gap between these two objects. So while it might be obvious for us to say that's the nucleus and this is the background, but for the computer to do it, it's going to take a little bit more, or to make it easier for the algorithm to find that breakpoint between them if we do that little filtering step. And so I can use the default and click auto, and it will pick a selection like that. Now that does a pretty good job. There's a little bit of gray around it that may be the nucleus, it may not be, but it's done a reasonable job at selecting the object from the background. Now I use the default one here, but there are a whole heap of different algorithms in Fiji. And so if we do those same steps, that's what the output would look like when you hit apply. Using the default, it picks 847. But if we use the triangles algorithm, it takes a point of 460. So it just picks a different value along here. And this drop down here has a whole heap, and you can read the different papers that were, describe these algorithms, but they're, some derivative of finding peaks or finding maximum points and then picking something between them. And so using the automatic algorithms means it's more robust, and it will do the same thing over and over for you. Whereas if you're opening images and going, oh yeah, that's kind of the nucleus selected now, and then applying it, you might be biasing different things, subconsciously when you are looking for your control versus your treatment and your cells are shrinking or something. And so having it just automatically do it for you is more robust and a better way of doing it. And so you can run all of the threshold algorithms in Fiji by using this image adjust auto threshold. And you can see here that the different algorithms, most of them are picking roughly the same thing, but there's a couple that pick a lot more, and there's a couple that are looking for, the really bright objects relative to everything else. And so you can use that to your advantage with different steps in your analysis pipeline. But that's a nice little quick way to see all the different options and how that would apply to your images. Okay, so in this second exercise, there's two example images. One is the example ramp image, and one is some nuclei. So if you open them up and have a go at manually doing the thresholding and using some of those auto algorithms to threshold out the nuclei. As a warning, the auto algorithms don't really work with the ramp image because they're not designed for that type of thing. And so I will kind of slowly open up the ramp and do some examples, and you guys can work along at the same time. So,
hey Nick.
Yep.
So when you set threshold levels, were you saying to subtract 244 then, or sorry, I think I missed.
So once you
that part.
So we'll come to that.
Okay.
So if I just quickly show you thresholding, I'll do the nuclei one.
Okay.
And so if I make the brightness, we can see all of the nuclei. You could go and do a pre-filtering step. You see that's taking way too many with the default. So let's change it to the triangle. And so let's say that my analysis step would be something like, I've got these cells, and they also had like a green channel where I was imaging cytox to see if the cells were dying. And so I can do the threshold and count how many objects there are and go, oh, there was 100 cells, and how many of them are green. So I have to take these objects to my green image somehow. And if we look up on my status bar, these nuclei here have a value of 255. And we will do this as a later exercise. And if I wanted to get rid of everything that wasn't a nuclei in the green image so that I could just measure the nuclei and say what the intensities were, I could multiply my mask image by my GFP image, for example. And if you multiply anything by 1 or 0, the 0 will be ignored, so the background will be taken away, and only the objects that I'd be interested in will be kept as 1 to then go and do like the next step of the analysis. So that's what I was referring to with the math step there.
Oh, okay, thank you.
That's okay. So I'll open up the ramp image. And so I can do, the default algorithm will pick 50%, but that's just to show you. It's all what thresholding is doing. It's just defining some kind of level of gray that you want to keep and ignore everything else. Yeah, it's a concept that some people struggle with, and I kind of struggled with at the beginning, but once I found this example, I thought that explained it really well. So I kind of already went through that nuclei example, but just to show you, if you do the auto, it picks a lot. If we reset that and do a median filter, and then it made no difference. So why did that do that? I must have to do a background subtraction as well. That's better. So has everyone managed to do that? There's no questions coming up, no one's asking. We'll move on, shall we? Okay, so again, there's some answer slides with the steps. Now I've modified the PDF from the RDM cache, but I will put up the latest slides there as well, and they'll have these answers if you want to do the exercises later. Normally, if we have it face-to-face, I print them out, but we couldn't do that today. Okay. I mentioned before that color thresholding is different to normal thresholding in a RGB, in a single channel type image, or fluorescence images where they're separate channels. But you can use color thresholding if you have images like a H&E stain, and because they will come into the software as a 24-bit RGB image. You can go image adjust color threshold and open up this box. And there are different color spaces that you can deal with. You can deal with the classic RGB, or you can manage the color space in the hue, saturation, and brightness level. And so that's these color spaces. And you get these options when we open up this window. So you pick the right color space, and then your original or your filtered, and you have to adjust all these different thresholds for each channel. Now, what's easiest to do is to use the sample option. And so if you draw a little region on the image, you can say sample, and it will set all of these boxes up for you based on that area. And it gets you in the right kind of place to modify it slightly just to get the best for your selection. So in reality, you're going to get to have an image like this, where it's the H&E, and if you want to count all of the purple stained nuclei, then you can't normally just pull out purple in a multi-channel fluorescence image because that's a composite of two colors. And so you can adjust either based on the hue and select out the, purple and red range. Doesn't matter in the saturation level, and you don't want the really bright, you want the rest. And that would do it, make that selection for you. Even easier, you could do a little segmented ROI around it and do that sample. And so we've got the third exercise involves using the H&E stuff. Now, I appreciate not everyone here does this kind of imaging, but I like to teach what it can do, and then you can go back and Google it in 3 years' time when you have to do it. But it's a nice way to show what's possible and get people thinking about other experiments. So if we do this one, the idea would be to pull out, say, the purple area. And we do all the same steps that you were doing before: background subtraction, some median filtering, and things like that. And so I get my mouse back on that screen. I can bring up Fiji. We can get that sample image. Three, four. And so we can do background, head scale. So this is a light background image. It's only subtle what it's doing, but it will make your selections easier later on. Now, if I was to draw around that purple one, image, adjust, color threshold. I lost my selection, but you get the idea. You can do that sample. If we change our color space, we can go back to our original. You can adjust how much blue it's selecting. Obviously, it's going to need a lot of blue and red. Make purple. And then you can take things out like a select, and then it gives you the selection. You could then apply that to your ROI or invert it and fill the rest to do your counts. But it's not a common method that people use, but it is one that can be really useful with picking things in color images or non-classical fluorescence images. Is anyone here actually using bright field imaging or, colored imaging like that in tissue sections? You can get the names. I know some people are in the list, so it can be a nice way to separate them out if you want to do quantification. And what's nice is you can actually get the macro command and then feed that in to do it on like all your images, which will be the third workshop on advanced topics that we'll cover. But you can get selections from that. You can use it for bright field as well. It's a bit more tricky. Like if you just have phase contrast images and you want to select the cells, there are other methods that you can use and employ for that. So come and talk to me if you have questions on that kind of space. Okay. All right. Again, answer slide, so you can zoom in and do that. Draw a region, hit sample, or you can manually play with the sliders to select the purple region, do pop profile or something if you want to get the maximum counts, or you can do like what I did, where you just put the cursor over it and have a look at the status bar in Fiji. So once you've done your threshold, no matter which way it was, whether it was color thresholding or your normal thresholding of intensity, you then end up with what's called a binary image. And so that's that image that has 0 or 255 as pixel intensities. And so if we use the lovely ImageJ example here, you can perform lots of different modifications to your binary image. So if you take your original gradient image and perform a threshold, you end up with the binary. So it's all the same now. You can perform an erosion, which will shrink it in by 1 pixel. You can perform a dilation, which expands your selection out by 1 pixel. You can do an open, which does the erosion and then a dilation. And then the close, which will close holes, does a dilation, then an erosion. There's the outline, which will then take just the boundary. And there's skeletonize, which will do shrink all the way down like an erosion until there's only 1 pixel left. And so that can be useful for like tracing dendrites and things like that. There's also these other ones down the bottom here. The main one that people will use is a watershed. And the watershed will let you chop up objects. So 2 closely touching objects, like if you had 2 nuclei nearby, you can watershed and try to cut between them. It can be useful, but you should always be checking what your watersheded images look like, because it can chop up too much. So if you had like 2 cells that were oddly shaped, it doesn't really work. It works for nice uniform objects like nuclei, but you should always be checking what you're doing, or what the output is once it's being chopped up. Okay, so the last kind of step then is to get it from that binary mask into a big list of all the objects. So then you can go and measure them individually, or you can take the areas of those objects and go to another channel and measure them or something. And so you want to end up with particles, and the word particle here could be cells, bacteria, objects, basically, in your image. And so you may have seen on my screen that I brought up the region of interest manager. Again, this was covered in the first workshop, the basic one. But what it does is it lets you store regions of interest. So you could be drawing a square around objects or sub-regions of your image, or you could be tracing the outline of cells. Manually, you can just click add, and it will remember that location. So you can take one selection from one image over to another image, and it will know the coordinates. You can hit add, you can hit T, Commands T, Control T, it'll all add it to the list. Now it's pretty obvious, like most things in Fiji, if you pick on one of them and then change it, you can update the selection, you can click on one and delete it. If you haven't clicked on it and you click delete, you should get the option to delete everything. You can go and rename them if you really want. You can measure things on it. So if you click one and then hit measure, it'll measure the 1. If you have a list of 10 ROIs and you hit measure, it'll measure all 10. If you have a complex data shape, so if you've got like a time series, you can come down to more, and then there's the option multi-measure, and that will then measure for every frame of that same region. So you can use that to get more measurements out, basically. You can toggle these boxes, the show all and labels. So show all will show the outlines. So if you are manually drawing around cells, for example, I like to have show all on so that you know which ones you've drawn around and which ones you've missed. And labels will just label them 1, 2, 3, 4 all the way through. So that's the region of interest manager. But you maybe don't want to manually do it. You want to parse your selections automatically into there for you. And so that's when you use the Analyze Particles tool. And so you want to create this selection from your image. So you go from original nuclei, you defined the thresholds, and then you apply it. So you've got this 0 and 255. And then you can take it to the next step where you automatically have it trace around each one of these individually. And so, as I said, you use the Analyze Particles, which is under Analyze Particles toolbox, and you get this little pop-up. And this lets you set up the rules for what it's going to look for and output for you. You get the size option. So again, that's pretty straightforward. You tell it the area, in a range. So if you don't want to pick really big, badly threshold parts in your image, you would set an upper limit. If you want to ignore like a tiny thing, like little speckles of noise, you'd set a low limit to infinity size. And you can importantly also filter here via circularity. So if we have performed those measurements before where we knew 0 is not round, 1 is round, and we had a cutoff somewhere of, I think the rod shaped one was 0.66. So if we're looking for that, we could add, 0 to 0.8 or something. So we don't want the really round ones, we wanted the elongated ones to do that. Show is what you can output. So you can output a mask image of everything it found. You can do nothing. You can just output the selection it makes. Again, they're all pretty self-explanatory. The other options, display results, it'll measure them. If you already had stuff in the results window, do you want to get rid of that first? Again, pretty obvious. Summarize will give you a one-line box that says, I found 10 objects. The mean area was 50 pixels and, a few other measurements by default. Add to manager is going to add them over to that result region of interest manager for you. Now, we also normally would exclude things on the edge. So if there was like half a nuclei, we wouldn't count that because you can't accurately measure things that you don't see the whole object for. Including holes will include the, if there's like a hole in your threshold, it's like a donut shape, whether you want to measure just the object or everything. And the last two, you don't really need to ever use. So we've taken our threshold image and we want to decide what parameters we want to feed into it. And so there's this little blue dot down here, which is probably an infection in the cell culture. So we've got big cell nuclei and then this little, probably bacteria. And so what I've done here is I've taken the magic wand tool, zoomed in, clicked on it. So it's automatically found that area and I've measured it and it's got an area of 12. So if I set my rules here to be 15 to infinity, it's not going to count this little spot and it's going to count the other nuclei. And I want to display them, clear any results I already had, add it to the manager. Let's click OK on the picture. So then when you do it, you can see it's labeled these 1, 2, 3, 4, 5, 6. It's not added this one because there's only 6 here, and it's measured them and given me the results. And this is where that's all well and good on my threshold that it's measured it, but the mean intensity is kind of irrelevant to me because it's just said that, yeah, they're all perfect black because that's what you told it to be with the threshold. And so what you probably want to do is take these areas and then measure them in another channel on your image, or go back to your non-thresholded original nuclei channel and measure it to see if it was equally labeled. So you get these numbers in the results window. And so for the next exercise, we've got that nuclei image again, and combining all the steps that we've gone through. So you want to massage it a little bit, do that pre-processing. So perform a median filter, do a background subtraction, make your threshold. Then you're going to have your binary image, and you can see that some of these are combining. And so you perform a watershed to see how it chops it up, and then do the Analyze Particles and add it to the manager. So it's a bit more complicated, this one. So if we then bring up our threshold, does a pretty good job. Let's use the triangle so that we've got some touching ones. And I want to apply that now. And so if we go process, binary, watershed, you can see that it's chopped this one. It's chopped that one that was touching. It's not perfect because I think this is 3 nuclei. You can see that let's chop this one down here as well. It does a good job, a reasonable job. And then if we go analyze particles, display the results, clear them, show a mask of what we had. And let's do this. Okay. And we end up here with our image. So that was our original image, and that's our mask. So it's excluded these ones that are touching the edge because we don't know how big they are and we don't want to measure them. We can see that it's got 2 objects here, and I can bring up the ROI window, hiding in the background, and I can go through. And you can see as I select it, it changes color, so I can see. I can turn it all off if I'm not interested in those, you know, seeing where they all are. And I've got my results window, and I can see all the areas of those nuclei. Now I could save this out, or I could copy, select all and copy, and then go to Excel and, you know, perform my statistics on that. People have any questions with that one? And so again, there's the answer slide that goes through some of these steps. So in this example, I took the ISO data instead of the triangles, different algorithms that are doing most, you know, different levels of selection based on the intensity values. Okay. Another way that you can use in Fiji to segment or find objects is the find Maxima menu option. So it's in the process menu again, and it's good for creating a single pixel selection of high intensity values relative to the rest of your image. So the tolerance option that you get is basically how much does your object sit above the, you know, for your selection. So if you imagine drawing like a line scan, you'd see all the peaks. What's the difference from background to the foreground objects that you want to put in? So this is a 12-bit image, I think, or 16-bit. So if we want to pick these like nucleoli bits, then we could make sure that our noise tolerance is greater than what the rest of the nuclei is to the background, but more the difference between this and this object. And you can output things like a single point, and then you get them. You can also get counts. So if you had just nuclei, you can perform more of a median filter, so something like a median of 7 or 8, and blur all of them, and then just say find those peaks in the image, and it would tell you and give you a single line output how many peaks there were. And so, you know, slightly different use cases allow for making this type of selection. So in the exercise example, there's this really noisy image. My PowerPoint's really struggling for some reason. So there's these 2 bacterial colony images, and we want to find these big bright spots of bacteria in the image. And so the way we can do that is open the images, zoom in, and see what the measurement of the intensity would be in those kind of brighter ones, and then do that find maxima. So we blank that, come back to Fiji, finding maxima. So it's the same image that's either inverted or not as the light and dark options, depending on whether you want to do the light backgrounded image or dark background. So we zoom in on some of these bright ones, and if I'm looking up on my status bar in Fiji, you can see that, you know, the value here is about 190. The dim one, it's about maybe 90. That's 190, and these dim ones like 50. Everyone see the cursor? So if we zoom out and go process, find maxima. Now, if I preview it with just 10, it's going to find a lot, because that prominence of 10 isn't much above background. But if I put in something like 150, then it's only going to find those bright ones that we were looking on before, because they're 150 above the background. And so another way to show that would be, I come in, oh, I don't want that, and draw the land scan. We can see our background's hovering around here, but the bright objects are up around, you know, 100, 200 almost. And so you come from, find a peak that's above that amount. So it's a useful way for making kind of unique selections. And so if we do that, find maxima, preview it, yes, I can output like a single point. And then I get this image with single point. And as an example, I've used this method when we get really densely packed nuclear stain in tissue, and people want to see if their single cells are lighting up with their GFP marker or something in that tissue, or you know, so it's quite hard to threshold those really densely packed nuclei, but you can blur it and you'll get like little undulations in the intensity, and you can define what that above background would be and get these points. And then you can do something like, because this is just a binary image, you can go in and dilate those holes a little bit bigger, so then they're about the cell area, analyze the particles, and then go through and, you know, ask, is it red or green or whatever the stain was. So that's one use case for it that I've found. Another really good use case is if people are doing colony forming unit assays and sitting there and counting all the little spots. This works quite well if you bang the plate on a stereo, capture all the points, and then do this. So, all right. I'll just quickly show the light version, which is just inverted. You get that extra option. It's got a light background. Because obviously if you don't tell it it's a light background, it's only going to find like one thing that's that different, because it's expecting it to go from dark to light, not the other way around. And it will make the same selections with the same values. Okay. Again, answer slide in the PowerPoint or the PDF that's up there on the cloud, where you can go through and see what it's doing and the steps to do that. Okay, so I had the question before about the image, when you do that subtraction down from 255 to 1, and this is the step in which we'll go through and show where that can be useful. So you can perform image calculations in Fiji. So before, where we were doing like the math on one image and just taking away something, you can actually multiply two images together or divide one image by the other, because it's just an array of values. And so you can perform these arithmetic on entire images using the image calculator. And that's under image process, image calculator. And these are, so when you get the window, it pops up this, and you have image 1, and then the operation, and image 2. And so this is all the different algorithms. So if you add them, then it's going to add image 1 to 2. Subtract, they're pretty obvious. But some of these, if it's like and or in the image, you can do, they can be quite powerful with doing things like better watersheding or more custom watersheding. You can use the and window, and you can combine that with one of the outputs from the find maxima. So instead of single points, you can find the valleys essentially between the points. So it can be quite useful. What you are doing is you're performing the calculation on that whole array. So you have to ensure that the bit depth's equal, because there's no point dividing a 8-bit image by a 16-bit image. You want to make sure that they are equal. It might be useful for when you're making a mask. So as I was saying, you'd threshold out regions that you're interested in, and then if you want to take only that region on your other image, then you would multiply your mask or your image by the mask, and then get rid of all the non-foreground objects. You can do ratiometric imaging, so where you're dividing like fret and the donor image one by each other. You use the divide. If you're doing like flat fielding calculations, you can subtract or multiply by your flat field image. Now, some of the outputs, so if you're dividing one image by another, you want to probably go up to a 32-bit float image. So these have pretty much an infinite range of intensity values. So if you're imagining that you're dividing, you're going to end up with decimals. And so have the 32-bit, it just keeps that smoothness of your image, and otherwise you're binning into two kind of small discrete values that it won't work, or two larger bins, sorry. So in the first example, as I said, you'd be making a mask. So if we had these cells and we wanted to ignore in our quantification this non-transfected cell here and just take the ones that were green positive, we could threshold the green channel and then take the multiply the mask of just the green channel, and it would remove that other cell for downstream parts of the quantification. Now, obviously, you can't use this for getting rid of that crappy cell in your field of view for publication. That's not on, it's not allowed, but you can use it to feed into other steps of the pipeline. Where this can be useful is, you may have noticed when you perform background subtraction, it takes 1 or 2 seconds, whereas most other things in Fiji are almost instant. If you're doing this on 10,000 images, that step takes a long time overall. And so this is one way where you can quickly do a sort of background subtraction or removal to do other steps where it's faster. And if you save 1 or 2 seconds on every image over 10,000 images, it can be the difference of something like, running for 10 or so minutes versus hours or days. So it's a useful step. But the important thing is, when you get this mask or the threshold, it's going to be 0 and 255. So that's where you'd use the math subtract 254. So then this is 0, this is 1, and you would multiply that on this image. Anywhere that was 0 gets ignored, and only the values of 1 where it was green is kept. And so there's 2 example images in the exercise 6. And they are, I think, 1 cell and 2 cells that are green. And we can do that same step. So they are stacks though, so you should maximum project them when you do your mask, so that you can make sure you capture it in all Z, but then you can multiply that by your stack. So I've got a stack. It's got 3 channels, red, green, and blue. If I Z project it, and then split the channels, take just that green cell. Now, because I'm performing a threshold, I want to kind of do a median filter first, just so that when I do that threshold, I get the whole cell. That's probably a bit too much. So I could go through and have a look at the different algorithms. So that should do. Apply that. Now this is 0, that's 255. Process, subtract, 254. Now it'll look black, but when I'm watching up here, I'm at 0, and then where the cell is, it's 1. So I can rename this one my mask. And if I go process, image calculator, I can take the original image and multiply it by my mask. Now it looks at, I could probably dilate my selection or something. It depends on what my downstream application is. Perhaps I'm only interested in transfected cells that have taken up bacteria. So it doesn't look pretty, but it's sufficient to then move to the next step and then threshold in that little region and ignore all the non-transfected cells or something. So do it this way. Close all windows. Yes. And I think the other example was basically the same. Yes. So it has 2 cells that are green and ones up here that aren't. So it doesn't really matter which one you use in that scenario, but you can work through that in your own time. So there's the answers for this one, basically showing those steps. And hopefully you can appreciate that now that we're in this segmentation step, we're doing those pre-processing steps all the time, for every example. Now for the next file that we'll work on, it's going to be a FRET analysis. So when we do FRET, where you're doing like acceptor over donor, then or FRET over donor, you end up dealing with really tiny values. And so in Fiji, like you might be used to these classic monochromatic lookup tables. So just how we show or present the data. And I just want to use this to point out that there are other methods to show data or other lookup tables that you can use. So our image in single channels are just like Excel spreadsheets of numbers. And if the number is 255, Fiji is going to display white. If it's like 100 and something, it's going to be a gray value. If it's 50, it's going to be a darker gray value. And if it's 0, it's going to be black. And that's your classic grayscale monochromatic lookup table. Now, if you have a green lookup table because you've got 2 channels and you want to show that it's green, then you can have the same kind of scheme, but green, pure green is going to be 255, and then you have kind of increasingly darker green as you go down towards 0. You can invert that whole lookup table to go the other way, and that can be really useful for showing fine structures in images. And so you do see in a lot of publications people inverting the lookup table for one of the channels, and it can be quite useful. There are these multicolored ones as well. So where this is going from black to white or black to green, this one will go through from black through to blue to white. And in this case, in this thermal lookup table, you go from kind of like a bluey purple through to a white hot through purple and yellow and red. Likewise, the spectrum one goes through all colors of the spectrum. Now, usually these type of lookup tables are presented with fret images, because in fret or flim and things like that, where your values might be, your whole image might only have a range of 0 to 3, and you want to show the subtle differences across that range, then you would use these ones, or these lookup tables. You can't use these as a multi-channel image. You can't also display your green actin on top of this image. It doesn't really work like that. But they can be used to make or better display your images. And there's a whole heap of lookup tables in Fiji, and there are ones that other people publish, and you can make your own if you really want. If you do make your own, you have to define it, obviously. So that's the, careful. Don't modify a classic lookup table just so that your, black comes in at 50. Subtract the background and do it properly. But you can go into the Fiji lookup table folder and make a copy of one of the lookup table files. They're like a .lut, and you can modify it. It's just an Excel spreadsheet of numbers. There is this little editor as well, where you can click in numbers and set define this one as black, and then you want blue starting at 50 and red starting at something else, and you can apply it. You can then get a window of that lookup table and show it. I've had a few people ask me over the years, which is why I've included it here. Okay, so in the second, well, this is the last example file, exercise, we are going to do a fret calculation. And so you have this multi-channel image, and we're going to use fret to see if or where the fluorophore, or where fret's occurring, and it only occurs. This isn't a talk on how fret works, but if you're not sure, if the two dyes, in this case CFP and YFP, are close together, then they will it'll share the electron, and you will see an increase in fret. And so to calculate fret, there's the donor channel and the acceptor channel, and we're going to there's to calculate the fret ratio, there's the donor, acceptor, and there's a fret image, I believe. And then you divide the fret over donor. So, sorry, the acceptor channel it's the acceptor dye, but under fret conditions. And when you do that division, then make sure that you've set the result to be a 32-bit float image, and you'll see that the cell it's really noisy because the values are so low. So change the lookup table to something like the spectrum, or try a couple of them to see, and you should be able to see a hotspot of where there's more fret occurring and where this protein is active. I'm just going to move an original copy and work on an extra one. So I could straight away split the channels. I can also duplicate that channel once I've done the math, and take a threshold, turn it into a mask, I'll take my resultant calculated image, and I'm just going to mask out all of the background. Actually, I'll leave it as 32-bit float because I've got a 32-bit float image that I'm giving it as the input. Now that doesn't show me much, so I'm going to apply a lookup table. I think this is where you want something like the fire or the, I have it on my screen. Let's just go spectrum. And see that my range isn't very much. Well, hopefully appreciate if I wanted to bring up the my image, if I go image, lookup tables, no, sorry, color, show lookup. I can see what the range is for this one. And so the fret's in here at the boundary. Do people have questions of that one? So I've just done, take my raw image, divided one channel by the other, and I get this result that's quite noisy. I then did that masking step that I did before and rechanged the lookup table. Because I'm working on quite a small screen, I couldn't actually scroll down to get that lookup table. But you get an image like that, and that's where the fret could be useful. Now you could, when you did that masking step, do a median filter and get a bit smoother boundary. But yeah, that's how you do the fret calculations, or an image calculation where you use the dividing, and you really want to make sure you use that 32-bit float. If you don't, it will cause all sorts of problems with your image. And so that's the workshop. Really fast today. Normally it takes a few hours. So